So welcome to our third in the series of cannabis epidemiology, and today we're looking at teratology. That's a really big word for birth defects. And Dr. Reece, world expert in this area of population studies around cannabis, is again with us. And Stuart, why did you study birth defects?
Well, we're interested in birth defects for several reasons. I see a lot of drug-dependent patients, and collectively they use a lot of cannabis, and we saw all kinds of birth defects, even cancers in our patient population. And the question was, could this be related to the drug use? Could it be related to cannabis? The published literature is a little bit divided on this. There are a lot of small studies which don't show a signal for birth defects. So the question was that if we went to really large populations, like the whole of the US for 20 years, or all of Europe for 10 or 20 years, would we pick up a signal? And that was the clinical question. Also, there is a lot of evidence for cannabis genotoxicity and epigenotoxicity in animals, rabbits and rats, that sort of thing. And there's a lot of the best human models, preclinical models for human teratology is actually white rabbits and hamsters. And in those species, there was a frightening array of birth defects reported in the older studies. The question was, as cannabis becomes more common, are we seeing that in human populations or not?
Okay.
So that's why we did the study.
All right. So take us through your presentation.
Okay, so let's talk about cannabis and birth defects and what the links are. We're going to talk about America, Europe, and then the comparison between the two places. I should say that some of these slides we're going to show here are a little confronting. People may wish to avoid the next few slides. This is a picture from the internet called Blue Babies, Teratology of Fallot. And you see shunting in the heart produces the blue at the edge, at the periphery, when the baby cries. It's called a TET spell. Gastroschisis, where the guts hang out and they can die, and even the liver can die, which causes, of course, a dead baby, as been described in now 7 large studies in association with cannabis. And carefully, no brain is linked with cannabis in 2 of our papers there, but also 2 papers from the CDC, which are mentioned there. And the rate here is about double reported by the CDC. And this is holoprosencephaly, and these are some photos of children born with that. Let's start off talking about Europe. Now, this is a picture of the rate of birth defects by increasing tobacco use. So this is there's 95 birth defects in Europe were tracked at the time. So you can't fit them on one slide. You've got to have 2 slides. But the point is that as the tobacco use rises, some of these lines are positive, like severe microcephaly, fetal alcohol, the nervous system, hydrocephalus, polydactyl. Some of these lines are sloping up in the positive direction. And then you come down to the second page, and the lines are all negative, in a negative direction, more or less. When it comes to last month cannabis use, you can see there's far more of the lines slope up in the positive direction. Just with understanding these graphs, think of the relationship between tobacco and lung cancer, right? We all know that tobacco is linked with lung cancer. So as the tobacco use rises, you'd expect lung cancer to rise, and you're going to get a line upwards to the right like that. So these upsloping lines become the smoking gun in epidemiology that we look at to see if there's a signal. And as we saw before, some of these lines slope down, some of them are flat, but there's much more of a positive signal here with cannabis than there was with tobacco.
And we should clarify, Stuart, that last month cannabis use is like really regular cannabis use. It's not like, okay, we can have weekly cannabis use, which is measured in particular surveys, or you can have last year. If a person has used in the last year, they may have used it very little. If they're using it weekly, they're using it a lot. Last month use is still high use.
Yeah. So this is daily cannabis use. You can see, and now these lines are very strongly positive. Almost all the lines on the page are positive. And then this is the second page. But this is a much stronger signal that you're seeing overall than what you saw with tobacco, which is why it's important. So this is teratological syndromes. This is birth defect syndromes. This is multiple birth defects across Europe in space-time. You see France and Belgium feature there. Belgium's black there, Bulgaria there, Germany, France, Spain there, Belgium there, feature in those plots. And this is teratogenic syndrome. This is the bivariate plot. So here we're plotting two covariates at once, in the pink and purple where they're both high, and the green where they're both low. And you can see that Spain and France, the teratogenic syndromes rose together. As the cannabis rose, so did the teratogenic syndromes. And this is called the VACTERAL syndrome, which is a medical acronym. It means vertebral, anal, cardiac, tracheoesophageal. The trachea is the windpipe. Esophagus is the food pipe. Renal and limb disorders. And this is the pattern that developed. This is a univariate plot across Europe. And then this is the bivariate plot. And you can see that the low countries, see they were high there, Belgium and Netherlands there, France and Germany there. The low countries were red there or pink there in the case of Netherlands, purple there in the case of France. So this VACTERAL syndrome is very interesting because it's thought to be due to a blockade of, there's a morphogen called sonic hedgehog, and VACTERAL cannabis blocks it directly and also epigenetically. So it's interesting that should have had that pattern. Now, this is the summary by system at 2 years lag. So we're looking here at 2 years lag of exposure, and this is the systems which are implicated in these 95 birth defects tracked in Europe. This is a number of anomalies defined and found to be linked with cannabis by organ system, cardiovascular, central nervous system. This is the percent of anomalies by organ system. This is kidneys and bladder, face, CNS, brain. This is the minimum E values, and that's cardiovascular, CNS, the brain and the face. And this is the total negative p-value exponents. So this is the exponents of the p-values, cardiovascular, general, face, and CNS is what leads to.
So just go back a little bit, because some of our audience will not have seen the earlier videos, might have missed those. Just very quickly explain the E-values and the P-values.
Sure. Well, p-values, P is for probability, and in science, a p-value of less than 0.05 is thought to be statistically significant. And the E-values relate to causation, and E-values of more than 1.25 are thought to be causal. E-values more than 9 are thought to be very causal. That's the tobacco lung cancer relationship. So these exponents, this is the exponent of, this is e to the 100 and e to the 50. These are massive exponents. This is the total of the E-value exponent for these organ systems. These are massive p-value aggregates for all of these organ systems. So that's implying a causal relationship. Now, so this is additive model summary by substance E and P values. So this is the number of anomalies with elevated minimum E value. So this is the minimum E value. This is a lower 95% confidence interval, and the number of anomalies. These are different. This is cannabis, daily cannabis interpreted. This is income. That's last month cannabis resin by THC daily interpolated there. THC, cannabis herb, cocaine, alcohol, tobacco, and amphetamines. And the point is that the cannabis indices are at the top end of this scale. Then if we look at the elevated, the sum of the elevated minimum E value, this is the exponents. Again, THC, herb THC, and last month cannabis, THC resin, and daily cannabis interpolated, THC interpolated. They lead the charge. Alcohol's there, tobacco's there. There's the sum of the minimum E values, and the sum of significant P values, P-value exponents. Income there, cocaine there, then cannabis, herb cannabis, last month daily cannabis interpolated, then tobacco and alcohol and amphetamines. So the tobacco and this system, this is the P-value exponents, negative P-value exponents. are a more powerful correlate of birth defects than tobacco and alcohol, which are known teratogens. We know that tobacco and alcohol.
That's important.
Yeah, we know that tobacco and alcohol damage baby growth. Let's look at the US. Now, this is the same sort of exercise that we just saw in Europe. So you can see here, this is all the birth defects. US track about 64. And this is rising tobacco exposure again. And you can see here that several of them have positive regression lines, positive slope regression lines, and most are nondescript, some are a bit negative. Now, when it comes to the last month cannabis exposure, it's a different picture. There's so many more positive sloping lines and very few negative ones. Some are flat, but it's a different picture. There's a handful of 2.5 rows of positive slopes with tobacco, and there's about 5 rows of positive slopes there with last month cannabis exposure from the US.
Just go back over those two. So go back to tobacco and just let it linger on that just for a moment so people can get a fairly good idea. And then to cannabis.
There's about 2 lines of defects there that have positive slopes. And with cannabis, there's about 4 lines or maybe 5 lines.
And it's where those slopes are going up that there is some issue with cannabis, some correlation.
That's right. Now, this is looking at E values. We talked about E values before as being linked with causality. So this is the log of the minimum E value, the natural log. And these are all the birth defects, and these are the logs for the tobacco relationship. And you see this scale goes up to 6. When we come to cannabis, the scale goes up to 15. Again, it's the log of the minimum E value, the same thing. And there's so many more birth defects are linked now with cannabis. And then with THC, again, you see that this goes 1.5. You see there's all these ones on positive and there. So we thought it'd be interesting to take a look in detail at a couple of birth defects. This is called small intestinal stenosis or atresia. Stenosis, the small intestine is the small bowel. Stenosis is a narrowing, or an atresia is a lack of growth. It just doesn't grow. It just shriveled up, and of course that blocks the bowel. So an acronym for that is CISA. And
now, presumably, they can operate on a baby. When the baby is born, they can operate and they can do something for this condition.
That's right.
Okay.
So that's the space-time pattern of CISA across the US.
Just go back to that. The states within the US that have high cannabis use, people may not know which they are. And
well, you've got Alaska, Oregon, and Colorado there. Are featuring prominently. There's some irregularity with the other states, but those ones are prominent.
Yeah, so you're always looking to see whether there is a correlation between those high use states, and Oregon being over on the left above California, the one that is kind of chevron shaped on the side, Oregon above that. And it will figure each time.
That's right. So this is called a mixed effects model, which is a commonly used powerful form of multiple regression, which looks at each state separately. And you see that all these terms for cannabis in all these models are strongly positive. You see the highly significant p-values there, 10 to the minus 10. And then we do the geospatial linkage, so we can do space-time regression. And we see when we look at space-time, again, the cannabis terms are highly positive, as you can see there. And this is looking at more complex model of different cannabinoids. And again, strongly positive signals. This is CBD, THC, CBG, cannabidiol, cannabidiol. This is the E-value list from this CISA group. And as you can see, this is very high.
So back to causality.
57 E-values there above the 1.25 threshold. And then from that, we can conduct construct models. That's a linear model, a cubic polynomial model, and a generalized additive model, which fits the curve like that. And we can show that the effect at the higher end is a lot more than the effect at the lower end. That's what that shows. And then we can look at, this is obstructive genitourinary defect. So this is a blockage usually in the bladder. which blocks the kidneys, which causes kidney damage in the babies, and requires remedial surgery to correct. And you see Colorado is high there, Alaska is high, there's New York, that's New Jersey there. So this is a mixed effects model. Again, cannabis highly significantly positive, geospatial links. Again, the cannabinoid terms are very strongly positive, as you can see there in the geospatial regression. Got 47 e-values there. Some of them are infinite. There's a strongly associated one defect, this one, obstructive genitourinary defect, and 47 above 1.25. And this is the model, linear, cubic, and generalized additive model, like that. And here it's to show that what this shows is that as the cannabinoid rises, the effect becomes much greater. In fact, in this case, what we're measuring is cannabidiol. So this cannabidiol seems to have a strongly positive effect.
And that's the wonder drug that's healing everybody of every ailment under the sun at the moment.
So this is the E-values. This is the cubic polynomial E-values from these models. And you see these are very, very high. It's the linear model there, 1.11, which is not significantly elevated, but in the cubic polynomial model, these E-values are very high indeed.
And I was being sarcastic about cannabidiol, by the way.
Right. Now, I want to talk about why we're talking about, I mean, one question here is how can cannabis possibly be associated with so many different birth defects in different systems? I mean, it's not just that it affects the heart. It's not just that it affects the brain. It's not just that it affects the kidneys. It's not just that it affects the guts or the limbs or the chromosomes. But how does it actually achieve this damage across so many different body systems? So to look at this, we're going to look at this. This is called an epigenome-wide association study, or an EWAS, from this brilliant North Carolina group that lead the world. And it was coordinated studies in rats and humans. So it's a two species EWAS study. And the p-values for the teratology from the shot EWAS study, you can show, this is the page, this is the number of genes, and these are the p-values, the cardiovascular, heart, atrioventricular valves, the brain, neurology, all these different systems coming out in this. The epigenome is the software of the genes, what controls genes expression. And of course, the genome controls everything. So this is the CNS, this is the brain and spinal cord, the brain, neurology, synapse, cerebrum, neurons, and the eye, which is part of the brain. And in all these different systems, you've got all these strongly positive p-values in this EWAS. The cardiovascular system, cardiovascular heart, atria, atrioventricular valves, ventricles, vessels. Again, strongly positive systems. This is the number of genes implicated in these different things. Now, this is an interesting progression from this is an embryology textbook about embryology, studies how babies develop. And it talks of this as an angioblast, which is the first cell in the baby that's going to become an aorta, or it's going to become a blood vessel. Then sonic hedgehog gets triggered. Remember the morphogen we talked about, which triggers vascular endothelial growth factor, which is another morphogen, which triggers Notch, another morphogen, which triggers Ephrin B2, which forms an artery. So anything that interrupts this progression is going to damage arterial formation. And cannabis damages most of those links there. So this is sonic hedgehog, SHH. This is a summary. This is the sonic hedgehog has a signaling system. It's a complex system. And this is from the EWAS study. GLI3 is one of the main transducers of the sonic hedgehog signal from the cell membrane onto the genes. You see there's 185 hits on sonic hedgehog, and patched here is one of the main sonic hedgehog receptor.
So when you're talking about hits, what does that mean?
That's changes where the cannabis has changed the epigenome from the control to the exposed condition.
Got it.
And then this is notched. Remember we talked about that progression. This is some of the damages with notch. And then this is VGF and Ephrin. Remember we talked about this is identifying all the steps. sonic hedgehog, VGF, notch, and eprin, just as we said. This is from the EWAS study that's identified when we study cannabis in notch. Now, when we turn to the face, again, number of genes annotated, 47 genes whose software programs disrupted for face formation, sensory organs, 29, the eye, the ear, all of these. So there's lots of hits on face. Then the general, so the embryo, DNA, mitochondria, microtubules, the breast, white cells, muscles, bone, vertebra, and bone. And you can see the number of genes annotated, lots and lots of genes annotated for all these diverse tissues. And these are fundamental processes here. So you've got cell growth, connective tissue growth, embryonic growth, organism growth, morphogenesis of the embryo, which is how the baby actually forms, and differentiation of embryonic cells as they grow and mature into a fully grown baby. And these are fundamental processes here: DNA transcription and RNA transcription. This is how the information comes off the genes. The DNA is transcribed, the RNA is translated. DNA binding, DNA replication, chromosome assembly, chromosome synapsis. That's where the chromosomes join and cross over. It's how the mother and the father chromosomes, maternal and paternal chromosomes, come together and form a baby, which is like the parents but different. And DNA recombination, DNA synthesis. So these are fundamental processes of the DNA. The mitochondria is very important because that supplies the energy for the genome and the software and the epigenome. So we know that disrupting mitochondrial function directly impacts the genes and the epigenomes. The microtubules, you know, when the cells divide and they separate, those tubules are made of tubulin. They're tubulin polymers. So anything that disrupts the microtubules is going to disrupt axon function, which is nerves, and it is going to disrupt cell division by interfering with the mitotic spindle. And then you've got muscles and all these bone ones. So this is explaining to a large extent. And then this is the limbs that we talked about. Number of genes annotated in limb development. This is polydactyly, which is extra fingers and toes. Polysyndactyly, which is extra fingers and toes that are joined. Polydactyly, apoptosis of the limb bud cells, death of the limb bud cells, and limb morphogenesis in the embryo. You can see, so all these genes here are implicated in limb development. And the limb actually forms as a meshwork of these different morphogens, fibroblast growth factor and Wnt, and retinoic acid. There's contrasting gradients of these, and these morphogen gradients determine exactly how the limb is going to form. And here you can see retinoic acid and fibroblast growth factor form these contrasting morphogen gradients, which weave the function of the guide the development of the limb in all species, really. And you can see fibroblast growth factor and sonic hedgehog here guiding and directing limb development. So anything which interrupts all of these key morphogen patterns is going to clearly disrupt limb development. And here we've got bone morphogenetic protein, fibroblast growth factors here, sonic hedgehog, its receptor patch, and all these genes here which are involved. This transduces the sonic hedgehog signal in the cell, which go into forming the limbs and the hoofs and the feet of all these different animals. And this is a detailed breakdown of the genes. We talked about mice-1 and fibroblast growth factor and retinoids and the Wnt's. They're all mentioned here and here. And you can see there's multiple 37 hits with mice-1, 97 hits mice-2, fibroblast growth factors, 175 hits. The retinoids, a lot of hits there, 203 hits on the Wnt's and chromatinase. So there's multiple hits here explaining why limb development could be disrupted by cannabis.
So cannabis is like a blunderbuss, which is hitting everything.
Yeah, that's right. And this is the gastrointestinal system, so the guts. And this is bowel cancer, bowel tumors, small intestine, large intestine, the esophagus, liver and pancreas. This is liver carcinoma, pancreatic carcinoma. We've actually looked at the epidemiology of pancreatic carcinoma in detail. And we found that what's reported here at the epigenetic level is confirmed at the epidemiological level, patterns of disease that you see clinically. This is the bladder and kidneys. And again, lots of hits here, lots of genes involved. The body wall, we talked about gastroschisis and exomphalos in some studies are related. Lots of genes involved there. And then the chromosomes. This is all these basic chromosome functions, multiple genes implicated.
So all of these genes that are getting changed by cannabis use, are they going to, like those changes, can they be repaired back or are they beyond repair?
Well, that's a good question. That's a complex issue.
Over generations, I suppose is what I'm thinking.
Yeah, look, we might come back to that a bit down the track. There
Yeah,
are
okay.
slides which speak to that issue.
Yeah.
The answer is yes, yes, and no. It's a complex answer. It's
All right,
not.
let's not deal with it now.
Mm. So, as I said, we looked at USA and we looked at Europe. If there's really a causal relationship between cannabis and these birth defects, it's more than just an association. You'd expect there to be a lot of overlap between the two studies. And of course, that's exactly what we did find. Now, as I said, Europe tracked 95 defects at the time we did the study. They did in the US 64. So there isn't one-to-one correlation between the anomalies studied. But to the extent that there is overlap, there's an impressive amount of commonality between the two series, as you can see. So this is very impressive overlap. Now, this is looking at your question, Gary. This is cannabis intoxication. This is cannabis withdrawal. And the point is that some of these genes, it does overall improve going from cannabis intoxication to cannabis withdrawal. But some of the signals persist and are strong, and some emerge for the first time in cannabis withdrawal. So that's what I mean. It improves, but in some cases it doesn't. So this is the negative log base 10 and a p-value by system. This is the different systems here. This is by target. This is the different systems in detail by the target. And this is the number of annotations by the target in cannabis dependence and cannabis withdrawal. And you can see it's lower in cannabis withdrawal, but still highly significant. This is the gene numbers affected. Again, lower in cannabis withdrawal, but still very significant. And the negative log p-values in cannabis withdrawal. This is the p-values, the logarithm of the p-values. And so it is better in withdrawal, but still highly significant. In other words, the cannabinoid genotoxicity is highly significant and is expressed in birth defects, both in Europe and America, with a lot of commonality, a lot of overlap, really as much as could reasonably expected, given that the two lists of tracked defects are slightly different. So this strongly confirms that the birth defects is a real and genuine expression of cannabinoid genotoxicity in large data sets. And because the defects themselves are rare, you wouldn't really expect to see them in the small studies. There's a lot of small studies which report a null result. But that's a bit unsurprising given the rarity of these defects. But these large series show very clearly that cannabinoid genotoxicity expressed in birth defects is a major epidemiological phenomenon of neonatal epidemiology.
Okay, thanks very much for that, Stuart.